So when you talk about the cell cycle is just the entire lifespan of a cell, what it's doing over its lifespan. So when we talk about mitosis and the cell cycle, they're not one and the same thing. If we look at this diagram here from your textbook, if we look at mitosis in terms of the total life cycle of the cell, so the whole circle, the whole pie is the life cycle, we can see that mitosis is only 1/4 of that, right? So it's only a small piece of what a cell does. And if you remember when you looked at the whitefish blastula and the onion root tip, most of the cells were in what phase? What'd you say? Interphase, yeah. Interphase is when the cell is not actively dividing, it's not getting ready to divide, it's just hanging out and doing what it's supposed to do. Like our brain cells, very few of them undergo mitosis. We have some that undergo mitosis, and those are the ones that become cancerous and cause brain tumors. But the part of the brain cells, those brain cells that conduct impulses and help you to think and move, those don't undergo mitosis. So we know that if they're damaged by a stroke or an injury, they don't recover. If the whole cell is destroyed, we can't replace that injured cell. But we know if skin cells can recover, right? You cut yourself and you get new cells to replace that. Bone cells can replace themselves by mitosis, so that works. The digestive tract is lined with cells that undergo mitosis. So we have certain parts of our body that actively replace themselves, other ones don't. So that's why we have to take care of the ones that don't, like the brain and spinal cord. So when we look at the rest of this purple arrow, all of this is interphase. Interphase is broken up into 3 different phases, which we'll talk about. And each of these little areas before and after mitosis are checkpoints. So right after mitosis has completed, so it kind of goes clockwise around the clock, prophase, metaphase, anaphase, telophase. After it's divided, those new cells are checked for DNA damage because DNA polymerase does make errors, right? We talked about that last time. So if there's enough errors that have accumulated, then the cell is programmed to be attacked and destroyed by the body or within the organism, whatever organism we're looking at, and it would no longer survive. And that's good. That way we don't accumulate these mutations and damage DNA. So when a cell is destroyed or is programmed to die, that's called apoptosis. So the term, it's not apoptosis. It looks like, it should be read that way, but it's apo. The second P is silent. Apoptosis. So then that's the first checkpoint. And then as it continues to grow, and then if it's getting ready to divide again, it's going to replicate that DNA, because we don't need another copy of DNA unless we're going to enter into mitosis and form 2 new cells. So we're going to need 2 copies of DNA, 1 for each cell at the end of anaphase, right? But if it's not, then it's just growing and hanging out. So if it enters into the S phase, it's getting ready to undergo mitosis again. And then we have another checkpoint, the G2 checkpoint, and that's going to make sure that this process of replication has occurred properly and it's ready to enter into mitosis. So that's the other phase of interphase. But again, this G2 phase is only if we're going to go into mitosis and only if we have the S phase of DNA replication. And then we enter into the phases of mitosis. So there is a checkpoint in the middle of mitosis called the M checkpoint, and that's making sure that the spindle has formed properly and it's able to pull those chromosomes apart equally. Because the thought is, as women age, their eggs age, because the eggs in women in the ovaries are formed during the fetal development period. Before that mom is even born, those eggs are formed. So if a woman is in her 50s or 40s, those eggs are 50 and 40 years old. Where males in the testes, they're making sperm every 2 to 3 months, brand new sperm throughout the lifespan. So those sperm are not old. The eggs are. So what the thought is as the spindle develops and grabs chromosomes after fertilization and pulls the chromosomes apart, the thought is the spindle breaks and leaves chromosomes behind. And now that fertilized egg has an extra chromosome Or maybe if it was on the other side of the spindle breaking, it's missing a chromosome. And that's where we see a lot of chromosomal issues with people as they age. So there's a checkpoint to look for that kind of damage, and that will, again, prevent mitosis from continuing if the spindle is broken and you don't have chromosomes lined up in the middle, right? Talking about metaphase, right? Or pulling apart during anaphase. So interphase then, just prior to mitosis, the cell grows larger. So what phase are we in of the cell cycle if you look at your previous slide? The cells grow larger, the organelles double. What phase do you think that's occurring in of the cell cycle? It's just before mitosis. So if you look at the diagram, what was just before mitosis? Which phase? G2. So those first two bullet points are occurring in the G2, and then DNA is replicated. And what phase is that? Yeah, the S phase. So we see some growth in the S phase as well, but the growth and readiness of organelles for that's specific to mitosis, we see that finalized in the G2 phase. Okay, so I want you to think of the S phase is all about making DNA. So we have another copy to send to that new cell when the chromosomes separate during anaphase. Okay, so we saw in the diagram, most of the cell cycle is interphase, and that's when the cell's just doing its everyday routine. And that routine would differ, right? If we're talking about a pancreatic cell, like in the body, its job is to secrete insulin and glucagon digestive enzymes. So that's what it's doing during interphase. Where if it's a muscle cell, it's just contracting, right? Contracting and relaxing. So different cells have different functions. And we talked about nerve and muscle cells. Well, I talked about nerve, muscle cells as well. All the muscle cells you have at the end of puberty and growth are all the muscle cells you're going to ever have. So you can either increase their size or decrease their size by exercising. So we build more little protein units inside of our muscle cells to make them bulge and be strong and contract with force, right? And if you don't use them, then they shrink and they're small and weak. So everybody in this room has the ability to be a, maybe not super large, but to have well-developed, strong muscles. It's just a matter of working them, right, in the right ways, whatever your activity is. So interphase, made up of these three phases. Getting ready, the big thing about G2 is you're getting just ready for that mitosis to start again. So proteins necessary for cell division. DNA has already been replicated. And what does replicated DNA look like? We'll look at that in the next slide coming up here. So in mitosis is when the cells are dividing. So again, mitosis does not occur in brain or muscle cells, but in any cells that are getting ready to make copies of themselves. So during growth, nerve and muscle cells divide, right, before puberty, because kids are getting bigger and their brains are getting bigger, and so that would make sense that it would happen during that time. But when we look at animal versus plant cells, we see a couple of different things that are different. Do you remember there was that special tissue that we looked at in lab where we saw mitosis occurring? It was one of the test questions. What was the organism we looked at for the plant? What'd you say?
Plant.
Yeah, what was the plant? Yeah, the onion root tip. So the special name for the tissue that's found at the tip of the root that's growing is called meristem. So the meristematic tissue is where we find what we see at the onion root tip, and we see that scattered throughout the plant wherever there's going to be continued growth throughout the plant, right? Because we know leaves fall off of deciduous trees and we need to grow new ones. In order to grow new ones, you have to have tissue that is able to divide and undergo mitosis. So we find that in the root tip, and we also find it throughout the plant, so it can continue to grow and undergo mitosis. So in mammals, mitosis, like I said, when you cut yourself or you break a bone, that's mitosis. as an embryo grows to become a fetus, and as a fetus grows to become a young adult. That's all mitosis. So anytime, any form of growth is mitosis. So the key thing of mitosis is to divide these chromosomes that were in the original cell. So we have 2 cells, each with a set of chromosomes. So we have 23 different chromosomes, and we have a pair of each. One you inherited from your mom, one you inherited from your dad. We looked at alleles last time, right, in lab yesterday. So we have a total of 46 chromosomes in all of our cells, but in sperm and egg, when we go to reproduce, we need to make another human being with 46 chromosomes. So how many chromosomes do you think sperm and egg have? What? 23, half that number, right? So we're going to study in the next type of cell division, meiosis, is how we go from a 46 cell down to a 23 chromosome cell. And we'll talk about that later. So right now we're just talking about simple cell division, making copies of cells. So we have a 46 chromosome cell in a, let's say in a bone or skin cell. And at the end of mitosis, we have a brand new 46 chromosome cell that's an exact copy of the cell that was there before mitosis. So when an organism has one set of chromosomes, we say that it's haploid, or N. That's the term for one set of chromosomes. So sperm and egg are haploid. They have one set of 23 chromosomes, which are a mixture of mom and dad. And we'll talk about that when we talk about meiosis. But our body cells everywhere else has the full set, one from dad, one from mom. So those are what we say diploid, because we have 2 sets of chromosomes for a total of 46. So diploid means 2 sets of chromosomes. Haploid means 1 set of chromosomes. And if you look at the different organisms, the number of chromosomes doesn't mean it's more complex. For example, a plant, this particular plant, the southern adder's tongue fern, has 1,320 chromosomes. Seems kind of crazy, doesn't it, for a plant? And humans have 46. So it doesn't mean we're more complex. A goldfish, we've seen those. Those have more than we do, 94 chromosomes in a goldfish. So it does not determine complexity. So at the end of interphase then, when DNA is replicated, so this is one strand of DNA. What we started, well, I mean, when we look at DNA, how it exists, it's a ladder, right? But at the end of DNA replication, we have 2 new copies of DNA. So each of these is a molecule of DNA, and they're exact copies of each other. We looked at that, we looked at DNA replication. That's what your test was on, right? So each half of this chromosome, we call this, is called a chromatid, a sister chromatid, because they're exact copies. So I had 1 DNA strand, and now I'm going to have 2 DNA strands here. You have a double-stranded ladder, right? And that unzips and exposes the nitrogen bases like we looked at with DNA replication, and then we add another strand down each side. So we have 2 copies of DNA, and now those 2 copies are going to coil up and wind tight to one another and be connected in the middle by a structure called a centromere. So these are the 2 replicated chromosomes connected together by a centromere. And then they have these other special structures called kinetochores, which are going to help these chromosomes attach to the spindle and be pushed to the middle in metaphase. And then we're going to pull these 2 sister chromatids apart in what phase? Anaphase, right, in anaphase. So the goal is to separate these chromatids, put 1 in each new cell, pinch off the cytoplasm in the middle, and each one has a copy of DNA that existed in the original cell. So there's special organelles called centrioles that secrete the spindle, and it's a cage-like structure that is formed in early prophase, or I should say late prophase, more metaphase. So these centrioles, you can see they're starting to secrete the spindle, but they're going to separate and go to opposite sides of the cell so they can push the chromosomes in position to get them ready for mitosis. So I want to stop at this point now. I gave you the introduction to mitosis. Now I want you to look at the animation that I put on Blackboard, draw the phases, make four, each phase should have 4 quality statements of what's unique about that phase. And that'll be your assignment for Monday. Okay, so you can pick it up